to Chapter 11, Part 8, Fundamentals of the Nervous System and the Nervous Tissue. So in this chapter, we are going to learn about the very basics of nervous system. Why understanding of nervous system is important is it's relevant with related to the application of certain drugs and chemicals, as well as understanding of certain diseases with related to nervous system required the understanding of the very basics of its components. So nervous system is master controlling and communicating system of the body. The cells communicate via electrical and chemical signals. So because it needs kind of a quick processing, the signals are rapid, as well as because it's need to be very accurate, it's very specific. Usually it causes immediate response for a certain stimuli. So nervous system, even though there are lots of functions, you can classify them basically into three main ones, and they could be overlapping with each other. The first one is the sensory input. The information is gathered by sensory receptors from internal and external changes. We call them as stimuli. The second function is the integration, which means like the processing and interpretation of the sensory input is happening during the integration process. And the motor output is the third function, where the activation of effector organs such as the muscles and glands produce a response. The nervous system, we can divide them into two principal parts. The first one is the central nervous system, which includes the brain and the spinal cord of the dorsal body cavity, and it does the integration and the control center. And the second part was the peripheral nervous system, which includes the portion of the nervous system outside the central nervous system. It consists mainly on the nerves that extends from the brain and the spinal cord, and the spinal nerves arise to and from the spinal cord, and the cranial nerves arise to and from the brain. So in this figure, you would see the parts which included for the central nervous system, which is the brain and the spinal cord for the central nervous system, the cranial nerves, as well as the spinal nerves with its ganglions included for the peripheral nervous system. So let's go over some of the very basics of these two systems. So let's talk about the peripheral nervous system first. Peripheral nervous system has two functional divisions. The first one is the sensory, we called it as afferent division, and the second one is the motor, we call it as efferent division. So with related to the sensory or the afferent division, it further divides into two parts: somatic sensory fibers as well as visceral sensory fibers. With related to the somatic sensory fibers, they convey impulses from the skin, skeletal muscles, and joints to the central nervous system. The visceral sensory fibers, that is the visceral organs, so the impulses are from the visceral organs to the central nervous system. With related to the motor division, it transmits the signals from the central nervous system to the effector organs, such as the muscles and the glands. And it can further divide into two major divisions: the somatic nervous system and the autonomic nervous system. And the autonomic nervous system has two major parts too, as the sympathetic nervous system and the parasympathetic nervous system. So the somatic nervous system The somatic nerve fibers, they conduct impulses or the signals from the central nervous systems to the skeletal muscles. And of course, as we know, the skeletal muscles, they are voluntary controls. So it's a voluntary nervous system. Somatic nervous system is a voluntary nervous system, that is conscious control of the skeletal muscles. The autonomic nervous system, it consists of visceral motor nerve fibers. They regulate smooth muscles, cardiac muscles, and glands. And it's out of our control, conscious control. So we call it as involuntary nervous system. The two functional divisions are the sympathetic and the parasympathetic. They work in opposite way. So this chart actually shows or summarize all what we have talked about so far, the two major parts, the central nervous system, the peripheral nervous system. The peripheral nervous system further divide into sensory, motor, the motor further divide into somatic, autonomic, and the autonomic further divide into sympathetic and parasympathetic. Then let's talk about the histology of the nervous tissues. Nervous tissue consists of two principal cell types. They're neuroglia, we call it as glial cells, and neurons, which we already know, the nerve cells. So the glial cells, they're small cells which surrounds and wrap the delicate neurons, and the neurons are the ones which are excitable and that can transmit electrical signals. When we dig into the glial cells, there are four types of glial cells which supports the central nervous system. They are astrocytes, microglia cells, ependymal cells, and oligodendrocytes. So let's look into them separately. The astrocytes. They're the most abundant and versatile and highly branched of glial cells. So I have underlined highly branched, which means like it's very important. They cling to neurons and synaptic endings and capillaries. They have certain functions and they have been listed out in here. You may be able to read them and I will read out few. One is support and brace neurons, play a role in exchange between capillaries and neurons, guide migration of young neurons, and so on. So this is a picture of the astrocytes. As it mentioned, it's highly branched and it supports the surrounding neurons. The microglia cells. They're small ovoid cells with thorny processes and it touched and monitor neurons. So they migrate toward injured neurons and they can also transform to phagocyte microorganisms and neuronal debris because at the injured neurons, there's inflammation going on. You need to remove the debris. So microglia cells does that function. The other important cell is the ependymal cells. They can range in shape from squamous to columnar, and it could be ciliated. So the cilia beads to circulate the cerebrospinal fluid. They line the central cavities of the brain and the spinal column, and they do an important function because they form a permeable barrier between the cerebrospinal fluid in cavities and tissue fluid bathing the central nervous system cells. So as you can see it in this figure, they're composed of the cilia. The other type would be the oligodendrocytes. They also some branched cells. They process and wrap central nervous system nerve fibers, forming an insulator, which we called it as the myelin sheaths in thick nerve fibers. The neuroglia of the peripheral nervous system, they have two major types. This is peripheral. Earlier it was the central nervous system. Now it's the peripheral nervous system. The two major types, the satellite cells and the Schwann cells. So the satellite cell surrounds the neuronal cell bodies in the peripheral nervous system, and their function is similar to the astrocytes of the central nervous system. And the Schwann cells, it's also called as neurolemmocytes, and they surround all peripheral nerve fibers and form myelin sheaths in thicker nerve fibers. So it does have a similar function to oligodendrocytes in the central nervous system. It does have a very important function, and it's very vital to regenerate of the damaged peripheral nerve fibers. So in this figure, you would see the Schwann cells surrounding the nerve fiber, which forms the myelin sheath. So nervous tissue is formed with neurons, which we called as nerve cells, and neurons are the structural units of the nervous system. They are large, highly specialized cells which conducts the impulses. They have special characteristics. They have extreme longevity, that is, last a person's lifetime. So that's why sometimes when a nerve is damaged, if it could not repair, then the function is lost by the structure which is supplied by that nerve. It's amitotic with few exceptions. So it cannot divide. Cells divide with mitotic process, the somatic cells, but nerve cells cannot divide. So the early example which I mentioned before is attributed to this amitotic thing, because if a nerve is damaged, it cannot be divided again and make a new cell. So the patient could lose the function of the organ or the structure which is supplied by that nerve. It has high metabolic rate, requires continuous supply of oxygen and glucose. Of course, it conducts impulses, it should be rapid, and that process needs lots of energy. So it does have a high metabolic rate. All have cell body and one or more processes. That's another feature of the neurons. Neuron cell body, also called as the perikaryon or soma. Soma is referred to the name body. So biosynthetic center of the neuron is the cell body. And why we call it that is it synthesizes proteins, membranes, chemicals, and it does have rough endoplasmic reticulum. The cell body contains spherical nucleus with nucleolus, and some contain pigments. And in most, the cell body plasma membrane is part of the receptive region that receives signals and information from other neurons. And most neuron cell bodies are located in the central nervous system. We call cluster of neuron cell bodies in the central nervous system as nuclei, and the cluster located in the peripheral nervous system, we call them as ganglia. And you will encounter these names as you read the chapter. So what are the neuron processes? They are arm-like processes which extends from the cell body. Central nervous system contains both neuron cell bodies and their processes, but the peripheral nervous system contains mainly the neuron processes itself. So there are certain words we refer to. The bundles of neuron processes in the central nervous system, we call it as tracts. The bundle of neuron processes in the peripheral nervous system, we call it as nerves. There are two types of processes, dendrites and axon. So this is the structure of the motor neuron, and you would see the some of the structures which we have called. So this shows the cell body, the nucleus, the nucleoli, and the nerve fiber over here. So this is the axon part. And the dendrites, this is a receptive region, they're highly branched, and these parts depicts the dendrites. So dendrites, in the motor neurons, it can contain hundreds of these short tapering branched processes, and it contains same organelles as in the cell body. So it's a receptive or the input region of the neuron, and toward the cell body as a graded potential. So we were going to learn what is graded potential. It's also called as the short distance signals. In many brain areas, the finer dendrites are highly specialized to collect information, and they contain dendrite spines, which we refer to as the appendages, with bulbous or spiky ends. Then the axon. The axon structure, each neuron has one axon which starts at the cone-shaped area. We call it as the axon hillock. So the cone-shaped area is the one which we called as the axon hillock. And in some neurons, axon could be shorter or it could be absent. In others, the axon comprise almost entire length of the cell. So long axons are called nerve fibers, and axons have occasional branches called axon collaterals, and at the end, the axons branch profusely at their end, and it could be range from 1,000 to 10,000 terminal branches. So the distal ends are called axon terminals or terminal bottoms. The functional characteristics is the functional characteristics of the axon are they conduct impulses which are received from the cell body. So axon is the conducting region of the neuron. They generate nerve impulses and transmits them along the axolemma to the axonal terminal. And at the end of the axonal terminal, they secrete messengers. We call it as neurotransmitters, which release to the extracellular space, and it's been received by the next nerve cell. So the neurotransmitters can excite or inhibit neurons in contact. So the axons carries on many conversations with different neurons at the same time. The axons rely on cell bodies to renew proteins and membranes, and they can quickly decay if cut or damaged. The functions, more functions are axons have efficient internal transport mechanisms. Molecules and organelles are moved along the axon by motor proteins or cytoskeletal elements. The movement of these elements can occur in both directions. There are two types: anterograde, which is away from the cell body, or retrograde, toward the cell body. So toward the cell body, examples would be organelles to be degraded, signal molecules, virus and bacterial toxins, so on. So what is the clinical significance of this nervous system and certain things? So certain viruses, bacterial toxins, they can damage this retrograde axonal transport, and we have seen that in certain diseases such as the polio, rabies, herpes simplex virus, and certain tetanus toxins. These in these diseases, the causative agent will damage these transport mechanisms. Let's talk about the myelin sheath. The myelin sheath composed of a whitish protein lipid substance. The function would be it protect and electrically insulate axon. It increase speed of the nerve impulse transmission, and the fibers, based on the presence and the absence of the myelin sheath, we can divide them into two types: the myelinated fibers or the non-myelinated fibers. So in the myelinated fibers, the segmented sheath surrounds most long or large diameter axons. In the non-myelinated fibers, they do not contain sheath. So in that case, they do conduct impulses more slowly. In the myelinated fibers, they do conduct impulses very fast. The myelination in the peripheral nervous system, it is formed by the Schwann cells. The Schwann cells wraps around the axon in jelly roll fashion, which is one cell forms one segment of the myelin sheath. And so, which means like for one nerve fiber, there could be several Schwann cells wraps around the nerve fiber and form the myelin sheaths. The outer collar of the perinuclear cytoplasm, formerly we call it as the neurolemma, is the peripheral bulge which contains the nucleus and the most of the cytoplasm of the Schwann cell. And the plasma membrane of the Schwann cell, they have less protein, no channels or carriers, so it works as a good electrical insulator. The interlocking proteins bind adjacent myelin membranes when it's wrapping around the nerve fiber. So this is what it's been shown or what is being discussed so far, which is like one Schwann cell, they wrap around each other, wrap around the nerve fiber and form a very good insulator around this nerve fiber. So the myelin sheath, because one cell forms one myelin sheath, and if you see, it's been very segmented. So these are one Schwann cell, second Schwann cell, third Schwann cell. Therefore, in between these Schwann cells, there's a gap. There are areas which have not been wrapped around. So these are called the gaps, and we call them as myelin sheath gaps. They're located between adjacent Schwann cells. That's those sites where axon collaterals can emerge in these Schwann cell gaps. And previously, when you read old books, you might find the word nodes of Ranvier. So they're formally called nodes of Ranvier, but currently we use the word myelin sheath gaps. In the non-myelinated fibers, they're thin fibers. They're not wrapped in myelin, surrounded by Schwann cells, but no coiling. And one cell may surround 15 different fibers. The myelin sheaths in the central nervous system. Previously, we talked about the peripheral nervous system. So when you read or look at the slides, you have to look whether it's talking about the peripheral nervous system or the central nervous system. So that's why I highlighted in red here, the myelin sheaths in the central nervous system. They are formed by the processes of the oligodendrocytes, not from the Schwann cells, not whole cells. So each cell can wrap up to 60 axons at once, and the myelin sheath gap is present. The difference is no outer collar of the perinuclear cytoplasm is present in the myelin sheaths in the central nervous system. The thinnest fibers are unmyelinated but covered by long extensions of the adjacent neuroglia cells. And the regions of the brain and the spinal cord with dense collection of myelinated fibers, we used a special name, the white matter. and the neuron bodies and the non-myelinated fibers composed the gray matter. So let's look at the classification of neurons. The classification could be due to two major things. It could be a structural classification or functional classification. With related to the structural classification, there are three types grouped by number of processes: multipolar, three or more processes, one axon, others would be dendrites, and this is the most common and major neuron type in the central nervous system. The bipolar, it's the two processes, it's one axon, one dendrite, and it's a rare type if you find bipolar, and it is existing. It could be seen at retina and olfactory mucosa. The unipolar, one T-like process, that's two axons, also called as pseudo-unipolar. It's peripheral process associated with sensory receptors. The proximal process enters the central nervous system. So in your book, the table 11.1 shows the comparison of the structural classes of the neurons. You would see the multipolar neuron, the bipolar neuron, and the unipolar. These are the structural classification, and it's been nicely laid out in this table, the differences. So the functional classification, it's three types. The neurons can be grouped by the direction in which nerve impulses travels relative to the central nervous system. The sensory, motor, and interneurons. In the sensory, it transmits impulses from sensory receptors towards the central nervous system. Almost all are unipolar, and cell bodies are located in the ganglia in the peripheral nervous system. And the motor neurons, they carry impulses from central nervous system to the effectors. They are multipolar neurons. Most cell bodies are located in the central nervous system, except some of the autonomic neurons. The interneurons, they are also called as association neurons. They lie between motor and sensory neurons. They shuttle signals through central nervous system pathways. Most are entirely within central nervous system, and 99% of the body neurons are interneurons. So further about the interneurons, they are also called association neurons, and that's the same things that we have discussed before. So this is the end of the slideshow part 1, and you would need to definitely read the more information about the basics from your textbooks.